Hello students, I am Dr. Rahul Kaushik. Today we are going to cover another important topic of our syllabus, and this is industrial production, estimation and utilization of phytoconstituent. So today we are going to discuss the phytoconstituent Taxol. Taxol, you must have heard the name Taxol, but I like to tell you that Taxol is nothing. Taxol is basically a trade name of an anti-cancer drug, Taxol. So Taxol is the trade name of Taxol, which is an anti-cancer drug. This Taxol is obtained from the yew tree. If you can see the old tree of yew, this is the yew tree, Pacific yew tree. It is found in America. This yew tree, the ancient yew tree, is used for extraction of your anti-cancer drug Taxol, or you can say the Taxol. So Taxol, if you can see the structure of Taxol, it is the ring, it is the basic nucleus or the basic ring of your, it is the basic ring of your structure, which is called as taxane ring. So this complex structure, anti-cancer structure, is obtained from the Pacific yew tree, and Taxol, again I am repeating, it is the brand name of the company BASF Anti-Cancer Shaft for its anti-tumor or anti-cancer drug Taxol, and the Taxol is a generic name for Taxol. So it is very important to know that Taxol is the generic name, whereas the Taxol is the brand name of the drug. Introduction: if you look at the structure, it is a diterpene pseudoalkaloid which is obtained from the bark of Taxus brevifolia, Taxus baccata, Taxus wallichiana juk. This Taxus wallichiana is the Himalayan yew. This wallichiana juk is the Himalayan yew or the Himalayan variety of yew tree, and the plant yew tree, it belongs to the family Taxaceae. The Taxol, it occurs in the form of needle-like or fine powder. It consists of taxane nucleus, as we have discussed in the earlier slide. It consists the taxane nucleus. This is the taxane nucleus or the taxane ring, which is the basic ring system of Taxols or Taxols. So the Taxol, it is native to Pacific Northwest of North America and to Canada. The variety which is grown in Canada is called as Taxus canadensis. So we have various places for cold places, temperate places for the cultivation of this yew tree, and normally this yew tree is found wild in the temperate climate zones. If we look at the solubility of this Taxol, it is soluble in acetonitrile, methanol, dimethyl sulfoxide, but it is insoluble in water. Again, it is insoluble in water, and its main use is anti-cancer. And you can see the Himalayan, beautiful view of Himalayan tree, Himalayan yew tree. These are the yew trees of Himalaya. Cultivation processing of Taxus, if you can see, these are obtained from the bark of this tree is removed to get the raw material for isolation of Taxol, and these are the spiny leaves of your Taxol plant, and these are the fine flowers which gets maturated into fine fruits, small fine pine like fruits, and this is the final isolated fine crystals of your Taxol, not Taxol, Taxol. Taxol is the name for the powder. If we look at the mechanism of action, since the phytoconstituent is anti-cancer, so it should show some anti-cancer actions. If we look at the anti-cancer action, you can see this is the normal mitosis process. This is the normal mitosis process where in metaphase the chromosomes, they are arranged at the center, and through the spindles at the center, through the spindles from the poles, these chromosomes, they are divided into two parts, and this is the normal process. And these spindle fibers, they are made up of tubulin molecules, they are stacked on one another to form a mitotic spindle. In normal condition, these spindle fiber, they form and they degrade during normal mitosis. But what happen when because it is a uncontrolled growth of cells, so to control that particular growth we have to use some anti-cancer drug. And how this anti-cancer drug or paclitaxel is going to act, what it do, in metaphase, these chromosomes, they arrange themselves toward at the center of the cell. Unusually stable tubulin molecule stack and fail to depolymerize. So, what we have discussed in this slide, that spindle fiber, they form and they degenerate for normal cycle. But what happen when you use a paclitaxel, what paclitaxel do, this paclitaxel, it stabilizes the tubulin molecules in such a way that they are able to form, but they are unable to depolymerize. So, in this way what happened, this the transfer of metaphase to anaphase was blocked. So, in this way the cell mitosis is blocked and your cell remains in the metaphase frozen state, your cell remains in the frozen state in the metaphase. The cell cycle is arrested in G2 and metaphase of the mitosis. So, this is the mechanism of action by which the anti-cancer drug paclitaxel is going to work on your body. If you look at the production part of the paclitaxel, you can see that we use the dried bark of our taxane plants or taxane trees. The dried bark, it is treated with N-hexane for 24 hours. The purpose of adding N-hexane is to remove non-polar compounds which are generally having no pharmacological importance. The defatted material we have obtained after 24 hours, it is then extracted with ethanol for 24 hours at 37 degrees Celsius and filtered. After that extraction, the filtrate is vacuum evaporated to the residual form. To the residue form, we add the 15% lead acetate solution. The purpose of adding lead acetate solution is to remove the tannins and phenolic compounds which are settled at the bottom when we centrifuge the mixture. Okay, we filtrate the vacuum. The filtrate is vacuum evaporated to the residue, and to the residue we add 15% lead acetate to remove the tannins and phenolic compounds, and the mixture is then centrifuged at 500 ounce per minute for 10 minutes. So, what happened after 10 minutes, the tannins and polyphenolic compound, they settled at the bottom of centrifuge tube and you can utilize the supernatant liquid. The supernatant liquid is collected and evaporated to dryness. That dried residue is added with water and the mixture is extracted with ethyl acetate 3 times. After extraction with ethyl acetate for 3 times, the ethyl acetate fractions we obtained, the 3 ethyl acetate fractions we obtained are then combined and concentrated under vacuum using rotary vacuum evaporator. The defatted material is then extracted with ethanol for 24 hours. Actually, this is the repetition of the last step. This is not included in this. So, come on to after this we should take this step. The residue is then dissolved in methanol for further purification and subjected to column chromatography in a column pre-equilibrated with chloroform. It means that the column is prepared in chloroform and the silica gel column packing was done in chloroform. The elution from this column was carried out using following solvent systems: chloroform acetonitrile mobile phase in following fractions, like following ratios. First we use the 100% chloroform, then we use the 75% chloroform plus 25% acetonitrile, then we use the 50% of chloroform and acetonitrile, then we decrease the concentration of chloroform to 25% and increase the concentration of acetonitrile to 75%, and finally we run the 100% acetonitrile through the column to obtain the fine crystals of our phytoconstituent Taxol. So this is the mechanism of, mechanism of isolation or the production for Taxol, or you can say Paclitaxel. Paclitaxel is the generic name of Taxol. So this is the GIF image, or this is the imagination of the column chromatography, how column chromatography works. So we have added the mixture of the drug, and through this column, on the basis of affinity of the component to the stationary phase, these columns, these different phytoconstituents, they get separated. Suppose this is the sample, this is the column or stationary phase. The sample starts to separate. The sample starts to separate. The blue component is collected first because it is having least affinity for the column. Yellow component collected next because it is having a medium affinity to the column, and lastly you can get the red compound which is having the most affinity for the stationary phases. So this is the column chromatography. This is again the column chromatography. You can see the different rates of flow through the column, component flow at different rates through the column, and this difference in rate of flow is dependent upon the interaction of stationary phase and the mobile component. If the compound is having more affinity to the stationary phase, then it moves slowly, and if it have least affinity, it moves at a faster speed. In this way you can achieve the separation of compounds through column chromatography. If we talk about the properties of Taxol, you can see the purified Taxol here, or Paclitaxel here. You can see the melting point of Paclitaxel is around 415 to 421 degrees Celsius. The boiling point of Paclitaxel is 957.1 plus minus 65 degrees Celsius at 760 mmHg. The solubility of Paclitaxel is in methanol, acetonitrile and DMSO, as we have already seen in the first introduction slide. Density of Paclitaxel is more than that of water, it is 1.4. Storage, if you want to store the purified Taxol or Paclitaxel, you can store it at a temperature of 15 to 30 degrees Celsius. Form, it occurs in the form of white crystalline or white crystals powder, or you can also have needle like crystals of Paclitaxel, and when we process or when you recover the Paclitaxel crystals, they get converted into the crystalline powder form. The water solubility, if you see, it is insoluble in water. The lambda max of Paclitaxel is around 227 nanometers in the ultraviolet radiations. If we talk about the estimation, separation or identification of the Paclitaxel, we have HPLC and TLC technique for achieving the separation or estimation of the Paclitaxel in the drug sample. So in TLC, if we talk about the TLC, we take the silica gel, pre-coated silica gel plates. Silica gel plates 60 GF 254. 60 is the size of the sieve through which the silica gel was passed. G is the gypsum, and F is the fluorescent reagent that fluoresces at 254 nanometers. And this silica gel shows fluorescence under UV radiations. The mobile phase for TLC analysis was used as a chloroform and acetonitrile in the ratio of 7:3, volume by volume. The flow rate is not applicable for TLC. Parameters, the detection of TLC, that when you developed the TLC plate, the detection of the sample or spots, the detection of the spots or the bands of Paclitaxel in the chromatogram can be achieved by spraying the TLC with vanillin sulfuric acid reagent. After spraying the vanillin sulfuric reagent, you have to heat the plate at 110 degrees Celsius for 10 minutes, and after that you should visualize the plate under UV radiations at short and wavelength radiations, and you can see the RF value of Paclitaxel on the TLC chromatogram at 0.59. The sample for preparation of for TLC application was as follows. 50 mg of the drug sample is dissolved in 10 ml of acetonitrile, and this is used as a stock solution, and from this stock solution we have taken 1 ml of stock, and it is dissolved in 10 ml of acetonitrile, and the spot was done on TLC plate. After this sample development, you should spot the appropriate quantity of the sample on the TLC plate, and the standard was prepared by dissolving 10 mg of paclitaxel in 10 ml of acetonitrile. Actually CAN is acetonitrile, it is acetonitrile, and this is stock solution, and from this stock solution 1 ml of stock is taken and it is dissolved in 10 ml of acetonitrile and is spotted on the TLC plate. So, in this way you can develop the TLC plate and you can visualize it using vanillin sulfuric acid reagent, and the RF value of paclitaxel was found at 0.59 RF value. So, if we talk about the HPLC analysis, we have used the Novopac C18 column for HPLC analysis. Thus, mobile phase for HPLC analysis we have used is acetonitrile and water in a ratio of 40:60 volume by volume. The flow rate through the column was maintained at 1 ml per minute. The injection volume was 20 microliter for HPLC analysis. The detector was set at 227 nanometer UV detector we have used. The retention time for paclitaxel in the chromatogram of the HPLC was 15.1 minute. It means that after injection of your sample, the peak of paclitaxel in the chromatogram was appear at 15.1 minute. The sample was prepared for HPLC analysis by dissolving 1 ml of methylmalonic extract, and it is filtered, and the filtrate is treated with internal standard. Here internal standard we have used 50 microliter internal standard we have used, and the internal standard used is cinemaldehyde in the concentration of 0.2 microgram per ml. Similarly, for standard also we have used the variable concentration of paclitaxel along with the internal standard cinemaldehyde. So, this is the condition, these are the conditions for detection of paclitaxel using HPLC, and you can detect the peak of paclitaxel in the HPLC chromatogram under these conditions after 15.1 minute. So, this is the HPLC instrument we have discussed in our previous lecture also. So, this is the sample holder, this is the solvent tray, this is the quaternary pump, and this is the column holder. Here you can place the column, and this is the detector. So, in this way you can carry out the HPLC analysis of your drug sample. We can also do the HPLC analysis. For HPLC analysis we have performed the same techniques as in TLC, but the mobile phase used here is chloroform methanol in the ratio of 90:10 volume by volume. The TLC chamber saturation time was 10 minutes. The total run time for TLC development was 25 minutes at room temperature, that is 27 degrees Celsius. The mobile phase run distance was 8 centimeter from the point of application of the spots. The derivatizing reagent for HPLC analysis was vanillin sulfuric acid, 1% we have used. It means that 1% vanillin we have used for 1% vanillin is dissolved in sulfuric acid to develop the derivatizing reagent. The TLC plate was dried in oven at 105 degrees Celsius. The sample was prepared for HPLC analysis by dissolving 1 mg of drug in 10 ml of methanol. The standard was prepared by dissolving 1 mg of drug in 10 ml of methanol. For HPLC analysis we have to specify the UV maximum or the detector setting. We have detected the spots under UV radiations at 235 nanometer, and the RF value for HPLC analysis of paclitaxel was obtained at 0.25. This is the RF value of paclitaxel. This is the machine, HPLC machine. It is very in HPLC all the tasks they are very much similar to the TLC, but here automation is used at every place. Automation is used at every place. So, as you can see that here we have used automatic sampler. It means that the sample on the TLC plate they are applied automatically using the sample applicator. It is very much similar to a printer that we use in our lab. It prints the spots to be separated on the TLC plate. So, it is the automatic developing chamber So you have to set the solvent specifications, and it automatically mixes the solvent when you place the solvent in the in these tubes. So after that particular solvent mixing, it automatically do the saturation of the chamber, and after chamber saturation, the chromatograph immersion device, device is used for derivatizing. Here we have no mechanism of spraying the reagent over the plate. Here we immerse the plate in the reagent, derivatizing reagent. Here is the TLC scanner. This scanner is used for visualizing the spot on your high wavelength or low wavelength ultraviolet radiations, and this is the visualizer. This carry out the all the calculations of your differentiated spots on the chromatogram of TLC. So every aspect or everything in HPTLC is carried out automatically. So here is involvement of very less involvement of human beings. If you look about the utilization pattern of Taxol, as we know that Taxol is an anti-cancer drug. Taxol is widely used in the treatment of breast cancer, it is used in the treatment of ovarian cancer, it is used in the treatment of lung cancer, bladder cancer, prostate cancer, melanoma, esophageal, as well as other type of solid tumor cancers. So it has got its use in various range or wide variety of carcinoma or cancers. It is also used in Kaposi's sarcoma. It is a special type of sarcoma. Taxol is primarily employed in the treatment and management of metastatic carcinoma of ovarian glands after the failure of follow-up chemotherapy. When chemotherapy fails in ovarian cancer treatment, this Taxol is the drug of choice for treatment of this ovarian gland cancer. And it also its advantage is that it is used in the metastatic stage of cancer. So it is generally the second last stage of cancer. You can use the drug in that metastatic stage of cancer. It is also used in the treatment of breast cancers, usually after the observed failure of combination chemotherapy for metastatic diseases. So Taxol has got lots of uses in a variety of cancers. So be it any kind of cancer, and especially the drug is of especially the for solid tumors or the tumors which are solid in nature, it is the drug of choice. So Taxol is an anti-cancer drug. If we look at if we take a fast recap of our presentation, we can see that this is the column chromatography we have discussed. This technique, if you can see that we have put a mixture, and mixture is getting separated into different component on the basis of its affinity for the column, and you can see that the red part, it is eluted last. It means that this red part is having the maximum affinity for that particular stationary phase, and you can see the blue part which is getting eluted first. It means that it has nothing to do with the stationary phase, it has no affinity for stationary phase, and it is moving just along with the mobile phase. So it is eluted first, and the compound yellow compound which is eluted in the intermediate, it is having the middle polarity or middle affinity for the stationary phase and the mobile phase. So in this way you can carry out the column chromatography. You can carry out the isolation of the phytoconstituent using column chromatography. You can also do the purification of your phytochemicals using column chromatography. This is another image, another animation. You can see that yellow compound, it is eluted first, then it is red compound, then at the least it is blue compound which is getting separated through the column. This same column, and in the same mechanism or in the same manner, the column of HPLC which we use functions. So what happens when you inject the mixture flows through the mobile phase, and along with the mobile phase this component moves, but the component which is having a high affinity for the stationary phase, it moves at a very low speed. whereas the compounds which are having very low affinity or very high affinity for the mobile phase or very low affinity for the stationary phase, they move first. Like you can see that the yellow compound, it is getting eluted first, after that red component which is having the intermediate affinity for the stationary column, and in the last we have the yellow component which is eluted at the last. So, in this way HPLC column they functions, and the separation is achieved of the separation of the component mixture is achieved. So, I am taking you to the this HPLC HPLC instrument. This is high performance liquid chromatography or high pressure liquid chromatography. What happen here, that the solvent, it is pumped through the pumps. There are various kinds of pumps. There are quaternary pumps, there are binary pumps. These pumps, they are connected, they are also connected with each and every machine, or each and every part is connected with the computer software, and this is also connected with the computer software. This column is also connected with the computer software, not exactly the column, the cabinet in which the column is placed. Actually, we have to maintain the temperature of this column. So, everything is connected with the computer, and this detector is also connected with the computer. So, what happen? You inject the sample, you are running the mobile phase through the column, you inject the sample. After injecting the sample, it is getting it get flowing from it started flowing along with the mobile phase. This is the stationary phase. Suppose we have three compound A, B and C. The compound C, it elutes first, it means it has least affinity for the stationary phase. Compound B elute second, it means that it has the medium affinity for the column, and compound A, if it has the least affinity to the column. It is very important to mention here that if the column is non-polar and the compound is also non-polar, then they have more affinity towards each other. So, the compound moves very slower, and if the stationary phase is polar and the mobile phase is also polar, the same situation again arises. The compound moves very slowly through the polar solvent. So, if the stationary phase is polar and the component is polar, if they are of same polarity, then the rate of flow of this component through the column is low, and if, or you can say the retention time is quite high. Okay. And if the solvent, if the stationary phase and the component, they are of opposite polarities, if they are of opposite polarities, then you have the low retention time, means that they are having a weak affinity of the stationary phase and the component. So, the component is not getting it is not getting in the it is not attaching to the stationary phase, it is just passing through the column, it is having least affinity through the column, and in this way it is eluted first. So, the RT value for component and stationary phase of opposite polarity is very low, or in other words you can say that opposite the nature of the stationary phase and the component, lower will be the retention time, and the retention time will be higher if the stationary phase and the component, they are of same polarity. So, in this way you can carry out the HPLC analysis of your sample. We have discussed the HPLC analysis, where we have used the UV maximum of 235 nanometers. We have used detector at 235 nanometer. We have got the RF value at 0.25 for our Taxol. This is the HPLC. HPLC is the sophisticated techniques where all the aspects of TLC, right from the sample application to the sample visualizing, or to the sample development, or to the you can say the chamber saturation, development of your chromatogram, everything is automatic. So, what is the benefit of this automation? This automation gives you the reliable result or reproducible results, be it Delhi, be it USA, the results for same analysis under same conditions will be same. And this HPLC analysis, it is the advantage of this HPLC analysis, that if you perform the experiment at any geographical location, the results will be reproducible. The RF value of the component will be same at every place. No matter, no involvement of the environment, no involvement of the manual, or you can say the no involvement or the interference of the human activities or the environmental factors. So these techniques, HPLC techniques, are the choice techniques for analysis of your herbal drug samples through the herbal drug research. Utilization, we have already seen that it is used in the treatment of various solid tumor cases, like treatment of breast and treatment of ovarian and lung cancer, and the treatment of your bladder, prostate cancer, in the treatment of melanoma, in the treatment of esophageal, as well as other types of solid tumors. So these are the various utilization patterns of Taxol. In the next lecture, thank you so much. In the next lecture, we are going to cover our next topic, that is podophytotoxin. In the podophytotoxin, it is also an anti-cancer drug. In the next lecture, we are going to cover our next topic, podophytotoxin. Thank you so much.